Hello ladies and gentlemen, welcome to my YouTube channel. Today we are going to revise Biology Paper 2 that was written in June 2023. I've paid attention to this question paper after being requested by a group of students who follows my tutorials on this platform. I hope it will greatly help as you are preparing to sit for your Biology Paper 2 on 7 November 2023. We are going to discuss Section A today and finish off Section B in the next session. So let us go to number 1. Section A, candidates are required to answer all the questions in the spaces provided on the question paper. Number 1A is reading. Fig 1.1 shows the structure of a palisade cell. On Fig 1.1, label with letter A where photosynthesis occurs, and then B where aerobic respiration occurs. So you should know that a palisade cell is a specialized cell where photosynthesis occurs. So photosynthesis occurs in the chloroplast. These are the chloroplasts. And then aerobic respiration, it occurs in the mitochondria. Mitochondria are the ones that are having these sausage-shaped cells. And then let us move on to part 2. Name the chemical of life that is found in part X and Y. So first we are going to label the part that is labeled X. It is known as nucleus. And then Y is known as cellulose cell wall. You should be able to know the examples of chemical of life. Examples of chemical of life, we have carbon, hydrogen, nitrogen, oxygen, sulfur, and phosphorus. These are examples of chemical of life. And then you should know that in X we are going to obtain DNA and RNA. So for DNA, they are made up of carbon, phosphorus, and nitrogen. So we are going to list those chemical of life as the ones that are found in part labeled X. So why is cellulose cell wall? It is containing long fibers of carbohydrates, including hemicellulose and pectin. So you should know that the carbohydrates are made up of carbon, hydrogen, and oxygen. So these are the chemical of life that we are going to find in Y. Let us move on to the next question. In part B, we are having a Fig 1.2. It is showing the structure of a cell that is involved in human reproduction. We are supposed to identify the cell that is in Fig 1.2. It is known as human egg or ovum. And then on part 2, we are supposed to give one structural difference between the cell in Fig 1.1 and the cell in Fig 1.2. So the cell in Fig 1.1 is an example of a plant cell, and then the cell in Fig 1.2 is an example of an animal cell. So you should know the differences between animal cells and plant cells. Animal cells, they don't have cell wall, whilst plant cells, they have cell walls. And then another structural difference is that in animal cell, the nucleus is at the center, whilst in plant cell, the nucleus is on the side. Another difference is that animal cells, they don't have plasmodesmata, whilst plant cells, they contain what is known as plasmodesmata. And also the animal cells, they don't have chloroplast, whilst the plant cells, they have chloroplast. And also the animal cells, they possess what is known as lysosomes, whilst the plant cells don't have, they lack lysosomes. In this case, we're supposed to just write one structural difference between the cell in Fig 1.1 and the cell in Fig 1.2. So the difference is that in Fig 1.1, the cell wall, we are going to have a cell wall, but in Fig 1.2, the cell wall is absent. Let us move on to part 3. Relate the structure of the cell in Fig 1.2 to its function. So we can see that this ovum, it is possessing this gel coat. That gel coating only allows one sperm cell to fertilize the ovum. The size of the egg allows more space for nutrients to be stored inside the cell. The cytoplasm contains nutrients to support the developing embryo. The cell membrane changes after fertilization so that no more sperm can enter the egg. That was all about number 1 of Biology Paper 2 in June 2023. Let us move on to number 2. Number 2A, Fig 2.1 is a graph showing the effect of temperature on the rate of an enzyme-controlled reaction. We are supposed to describe the shape of the graph. So as we increase the temperature, the rate of reaction is going to increase till it reaches what is known as optimum temperature. After that point, the increase in temperature is going to result in the reduction of rate of reaction. And then on part 2, we are supposed to state the term that describes the temperature at B. It is known as optimum temperature. And then on part 3, we are supposed to explain the shape of the graph between A and B. So between A and B, it is just below optimum temperature. There is increase in the rate of reaction as more heat is provided. Heat energy is transferred to kinetic energy. If the enzyme and substrate have more kinetic energy, they move quickly and are more likely to collide with each other with enough energy to form enzyme to substrate complex, so more reaction can occur. Let me repeat to explain again the shape of the graph between A and B. Between A and B, it is just below optimum temperature. There is increase in the rate of reaction. Heat energy is transferred to kinetic energy. Enzymes and substrate move quickly, colliding with each other with enough energy to form enzyme to substrate complex, so more reaction is going to occur. Let us move on to part 4. Explain how the temperature at C illustrates the protein nature of enzymes. So enzymes are temperature specific. After optimum temperature, further temperature increase is going to result in decrease of enzymatic activity, because high temperatures denature the enzyme activity. And then on part B, we are supposed to state in 2 other properties of enzymes. Enzymes are biological catalysts. Small quantity of enzymes catalyzes large quantities of substances. Enzymes are pH specific. Activity of enzymes depends upon the acidity of medium. Each enzyme is most active at a specific pH. For example, pH 2 is for pepsin. Enzymes are highly specific in action. One enzyme can only act on a particular substrate. That was all about number 2. Let us move on to number 3. Number 3a, Fig 3.1 shows a cross-section of a dicot root. We are supposed to name the part labeled A, B, C, D. So A is known as phloem, and then B is known as protoxylem, and this space at the center is known as metaxylem, and then the part labeled C is known as cortex, and then finally the part labeled G is known as root hair cells. Let us move on to part B. We are supposed to describe the functions of B and D. So we labeled our part B as protoxylem. It is the primary xylem in which narrow vessels are formed. It plays an important role in hydraulic function of vascular systems. And then the part labeled D is known as root hair cells. They aid in absorption of water and minerals from the surrounding soils. Let us move on to part B. Explain the effects of wilting on a plant. So wilting is known as kushava in Shona. It causes a decline of photosynthesis because the stomata closes, preventing carbon dioxide from entering the leaf. Wilting diminishes the plant's ability to transpire. Reduced transpiration means fewer nutrients for plant growth are going to be absorbed and distributed. As the transpiration decreases, respiration is going to increase, resulting in stored food reserves being reduced. Permanent wilting leads to the plant death. Let me summarize again the effects of wilting on a plant. It causes decline of photosynthesis because stomata close, preventing carbon dioxide from entering the leaf. Wilting is going to diminish transpiration, which means that fewer nutrients for plant growth they are going to be absorbed. It results in increase in respiration, reducing food reserves, and also the permanent wilting leads to plant death. That was all about number 3. Let us move on to number 4. Number 4a, state the function in the mammalian ear of the pinna. It is the visible portion of the outer ear. It collects sound waves and channels them into the ear canal where sound is amplified. And then we are supposed to state the function of auditory canal. It transmits sound from pinna to the eardrum. And then on part 3, we are supposed to state the function of cochlea. It plays a vital role in the sense of hearing and participates in the process of auditory transduction. And then on part 4, we are supposed to state the function of semicircular canals. The semicircular canals are very small tubes whose primary job is to regulate balance and sense head position or head rotations. Let us move on to the next question. On part B, we are supposed to suggest 2 effects of a very loud explosion to the ear. So it causes peripheral hearing impairment. It causes auditory processing disorder. It also results in vestibular impairment. And then on part C, we are supposed to explain how a person with hereditary deafness can be assisted. So we can use hearing technologies such as hearing aids. Also, we can use sign language and other means of sensory substitution. We can also use cochlear implants and middle ear implants. That was all about number 4. Let us move on to number 5. Number 5A, part 1. Name the substance that is found in the core of a virus. Virus is made up of a core of genetic material, either DNA or RNA, surrounded by a protective coat called capsid, which is made up of proteins. And then on part 2, we are supposed to state into diseases that are caused by virus. There are so many diseases that are caused by virus. Examples, we have AIDS, influenza, measles, coronavirus disease, chickenpox, human papillomavirus, Ebola, polio, hepatitis. So we're just supposed to state into diseases from all the diseases which I have mentioned. And then on part B, we are supposed to suggest why viruses are not considered as living organisms. Viruses are not made up of cells. They can't keep themselves in stable state. They don't grow. They can't make their own energy. They cannot reproduce on their own, and they require a host cell in order to replicate. Let me repeat to explain again why viruses are not considered as living organisms. They are not made out of cells. They can't keep themselves in stable state. They don't grow. They can't make their own energy. They cannot reproduce on their own and requires a host cell to replicate. And then on part C, we are supposed to state in 3 structural features of bacteria. So all bacteria, they have cell membrane to control the movement of substances in and out of the cells. They have cytoplasm, which functions for the growth, metabolism, and replication. They also have the ribosomes for messenger RNA translation and protein synthesis. Let me summarize again. Bacteria have cell membrane to control movement of substances. They have cytoplasm for growth, metabolism, and replication. They have ribosomes for messenger RNA translation and protein synthesis. That was all about number 5. Let us move on to number 6. Number 6A, define the term codominance. Codominance is inheritance pattern where both alleles in heterozygote are expressed equally. And then on part B, we are supposed to state into diseases that are caused by gene mutations. So first I'm going to explain what we mean when we are saying gene mutations. These are changes in the genetic sequence due to mistakes when the DNA is copied or due to damaging effects of mutagens such as chemicals. So the examples of gene mutation disorders, we have Down syndrome, sickle cell diseases, Huntington's diseases, cystic fibrosis, hemophilia, albinism. So these are the examples of gene mutation diseases. In this question, candidates were just required to state into diseases. Let us move on to part C. Name N2 factors that increase the rate of mutation. We have environmental exposures to mutagens such as tobacco smoke, ultraviolet light rays, X-rays, exposure to wide varieties of chemicals. Let us move on to part G. Part G, a man heterozygous for blood group B and a woman of blood group AB produced a child. Using a genetic diagram, show the probability of the child having blood group O. So first we are going to write the blood group of the father. So if it is heterozygous B for blood group B, it means that we are going to have a capital letter B and a small letter b. This means that it is heterozygous. And then for the blood group of the mother, we are going to have AB like this. And then we are going to distribute this so that it is going to be IB and I and a small letter b. It is going to be multiplied by this A and also this B. And then after that, we are going to do the genetic cross diagram. So let's say this B and this A meet. We are going to have a child with blood group B, A. So if we are going to have this B and this B meeting, we are going to have a child having homozygous B. Let's say we are having this IB and this IA meeting. We are going to have this blood group. And then finally, we are going to have this IB meeting with this IB in capital letter, so that we are going to have heterozygous B, which is like this. So you can see, guys, that it is not possible to produce a child with type O blood. The only way to produce a child with type O blood is for both the alleles to be O. The woman in this case only give an A or a B allele. So we are not going to have a child with probability with type O blood group. Let me repeat to explain again. The question was supposed to... So let me repeat again. The question is reading, using a genetic diagram, show the probability of the child having blood group O. So we have drawn the probability genetic diagram, and the probability is 0. It is not possible to produce a child with type O blood. The only way to produce a child with type O blood is for both alleles to be O. The woman in this case only give an A or a B allele. This marks the end of our revision today on section A of Biology paper 2 that was written in June 2023. Thank you so much, guys, for following me on this channel. Please don't forget to subscribe, like, and share my videos. This is Eve signing out. Let us meet again in the next session where we are going to discuss section B.